Hello, my dear Form 5 students. It's another time that we are meeting in our normal Elimika program. The subject we have at hand is biology. And the topic we have today is gaseous exchange and respiration. My name is Fortunatus Kyobe. As usual, welcome. Well, let us try to remind each other that our last session was about nutrition, and we had the review of what nutrition is, right from the beginning of the topic up to where we came to conclude the topic that was about hormonal and nervous control of the secretion of the digestive system. And we had some questions to wind up our topic. Now, the topic that we're going to start with today is gaseous exchange and respiration. I remember when you were in Form 2, you went through, though in an elementary way, went through gaseous exchange and respiration. Now, can you remember what gaseous exchange is? Can you remember what gaseous exchange is? How does it take place? In which parts of the body does this gaseous exchange take place? In both plants and animals. My dear student, you will agree with me that always we are taking in gases that are supporting our physiological processes. And the gas that we take in always from the environment, or the gases that we are taking in include all the contents of the atmosphere. Because when we are breathing in, we don't have a filter. We don't have a differentially permeable filter to take in or to filter out those gases which are required in the body, and to filter, to exclude those who are not required in our bodies. So we take in gases, we take out gases. But the process by which now respiratory gases are exchanged between the body cells and the surrounding environment is what we call gaseous exchange. Respiratory gases in this case, we have oxygen and carbon dioxide mainly. These are exchanged between body cells and surrounding environment. Now, the process of this gaseous exchange, well, differs as the mechanisms differing when we refer to gaseous exchange in plants and when we talk of a gaseous exchange in animals. In plants, for example, gaseous exchange takes place in different structures. There are different structures, there are different parts through which gaseous exchange is taking place. Mainly, gaseous exchange in plants takes place through stomata. through the stomata or stomatopores. When the stomatopore opens, oxygen for respiration purposes get in and the carbon dioxide get out. But at the same time, we have carbon dioxide for photosynthetic purposes getting in and oxygen as a result of photosynthesis coming out. Well, the stomata normally open during the day, and therefore we have the rate of gaseous exchange in the plants higher during the day than it is during night. But also, there are other factors that facilitate the rate of gaseous exchange. Temperature. At higher temperatures, the stomatopores get open. And therefore, the rate at which gaseous exchange is taking place at higher temperatures is also higher than the rate at which the same process is taking place when the temperature is low. Now, apart from the stomatopores, there are also other epidermal apertures. These are called lenticels. The lenticels, mind you, these are not cells. Don't take this last part to mean cells. Lenticels are not cells. They are rather minute pores, small apertures that are found either in the stem or in the fruits or within the leaves, which also, apart from carrying out other functions such as transpiration, they are also holes, they are also pores through which gaseous exchange takes place. But also, root cells. The root cells are also sites for gaseous exchange. And of course, these facilitate gaseous exchange between the plant cell and the soil air. The root cells are facilitating gaseous exchange between the plant cell and the soil air. Now we have a gaseous exchange in animals. Now, in animals such as mammals, gaseous exchange is normally preceded by breathing. Gaseous exchange is preceded by breathing. Mind you, please do not confuse between the two. Gaseous exchange and the breathing. Breathing means inhalation and exhalation. Taking in of respiratory gases and breathing out or taking out the products of respiration, that is breathing. But gaseous exchange, this is taking place across the cell surface membrane. When this gas from outside diffuses into the cell and the gas from inside the cell diffuses out. That is what we mean by gaseous exchanges. We're going to see in your preceding sessions. As I have said, that in animals such as mammals, gaseous exchange is preceded by. It is preceded by breathing. And this breathing involves inhalation, or we call it inspiration, and exhalation. or simply expiration. Now, when we inhale, the gas that we are taking in is a mixture of different gases. But also, when we exhale, that we are taking out is also a mixture of gases. Now, the composition of these gases differs in the inhaled as well as exhaled air. Now, suppose that we have something like a gas, we have composition in the inhaled air, and we have its composition in the expired air. We have different gases. For example, what is the composition of oxygen in the inspired air? You remember the composition of the atmosphere that we had in Form 1 chemistry? The composition of oxygen in the inspired air is always 21%. But when we breathe out, that we are giving out contains 16% of oxygen. When we breathe in nitrogen, for example, we take in 78% of the breathed in contains 78% of nitrogen. But when we breathe out, what we are giving out also contains 78% of nitrogen. We have carbon dioxide. When we breathe in, what we take in contains about 0.03% of carbon dioxide. But when we are breathing out, what is given out contains about 4% of carbon dioxide. But also we have water vapor. When we are breathing in, the amount of water vapor is variable. But when we breathe out, the amount of water vapor is concentrated compared to the amount of water vapor in the breathed in air, or in the air breathed in. Now, what interpretation can we give as far as this data is concerned? What interpretation can we give as far as these figures are concerned for oxygen? You can see that there is a decrease. There is a decrease of oxygen by 6% from 21% in the inspired air to 16% in the expired air. Why a decrease? Here we have some of this oxygen being used. Some of oxygen is used to oxidize the respiratory substrates. For example, you have glucose. This is oxidized by oxygen to form carbon dioxide, water, and energy. Now, the reduced oxygen is the one which is used in the oxidation of the respiratory substrate. Go for nitrogen. You see, nitrogen in the inspired air is 78%. But also in the expired air, we remain with the same 78%. Why is the percentage remaining the same? And this is because in our bodies, in our physiology, there is any process that involves the use of nitrogen. That's why this nitrogen content is remaining the same. We have carbon dioxide. Well, in the inspired air, we take in the air containing 0.03% of carbon dioxide gas. But when we breathe out, the air that we are giving out contains 4% of carbon dioxide. You can see that here there is an increase in carbon dioxide concentration or in carbon dioxide content for about 3.97%. The additional carbon dioxide comes from two sources. The first one is the result of oxidation of the respiratory substrates, as you can see here. But the second source is the decomposition of bicarbonate salts in the blood. One, carbon dioxide comes from oxidation of the respiratory substrates. As you can see here, glucose being oxidized to yield carbon dioxide, water, and the energy. But also, in the blood, there are hydrogen carbonate salts. So the decomposition of bicarbonate salts yields into the production of carbon dioxide. That's why we have an increase from 0.03% to 4%, a shooting of 3.97%. And lastly, we have water. We have water. You see, in the inspired air, the amount of water is variable. But in the expired air, the amount of water increases, so it's concentrated. Well, this depends on the humidity of the place. The amount of water vapor in the inspired air will depend on the humidity of the place. If the place is more humid, more water will be taken in. And the place is less humid, little amount of water will be taken in. But in the expired air, the amount of water is high. That is, there is a concentrated amount of water. This is because the additional water comes out as a result of oxidation of the respiratory substrates. And the amount of water vapor in the expired air will depend on the substrate that is respired at that particular time. For example, respiration of fats, respiration of oils will yield a substantial large amount of water compared to other respiratory substrates. This is because the number of hydrogen atoms contained in these lipids is higher than the number of hydrogen atoms that is found in other respiratory substrates. So we have respiratory surfaces. Respiratory surfaces in animals. Well, the parts of the structures that are used for gaseous exchange, these are referred to as respiratory substrates or respiratory organs. We have different respiratory surfaces or respiratory organs in different groups of animals. For example, we have lungs. Lungs, these are respiratory organs or surfaces of terrestrial animals, such as mammals, We have reptiles. We have amphibians, such as adult frogs and toads. Apart from the lungs, there are also gills. These are used by the aquatic animals. Example, fish. We have crustaceans, such as prawns, but also we have tadpole larvae. I mean, the toads and the frogs at their tadpole larval stage, they normally use gills as sites for gaseous exchange. We have skin, which is used by the adult frog when they are in water. But also, there are book lungs. Book lungs used by the arachnids, such as spider. But also trachea. Trachea, this is used by insects. Now, these are just a few examples of the respiratory surfaces or structures that are used for, that are used as organs for gaseous exchange. Yeah, so those are the respiratory surfaces. Now let's look at the features which characterize these respiratory surfaces, be it gills, lungs, trachea, or whatever. Provided that the structure is involved in gaseous exchange, it must have the following features or characteristics. One, that any efficient respiratory surface must have a thin epithelium or a thin epithelial lining. This to reduce the distance, to minimize the distance across which the exchanged gases have to move. The second one is that it must be moist. Moist in the sense that the gases that have to be exchanged, to be moved, that have to move in and out of the cells should move in their solution form. Now, the moisture on the respiratory surface is there to dissolve the gases so that they form solutions for easy movement in and out of the cell. The third feature is that any respiratory surface must be highly vascularized. Meaning that it is densely supplied with the blood capillaries to facilitate easy transportation of the exchanged gases. But also, any respiratory surface must have a large surface area. Large surface area for maximum loading and releasing of the respiratory gases. We are going to see soon, for example, the mammalian lungs, which features increase the surface area of the mammalian lungs. So those are the respiratory surfaces and their features. For example, a question might go that explain how the gills in fish are adapted to effect gaseous exchange. How are these gills adapted to effect gaseous exchange? Here are the adaptations. Now we have gaseous exchange in mammals, that is mammalian lungs. Well, the organs for gaseous exchange in mammals are the lungs. The organs for gaseous exchange in mammals are the lungs. And these lungs, of course, we have two lung lobes, and these two lung lobes are protected within the thoracic cavity, and protection is offered by the thoracic cage, which is made up of the ribs. And of course, between one rib and another is what we call intercostal muscles. Intercostal muscles. And on the abdominal side, the lungs are separated or are bordered by a muscular diaphragm. Around each lung lobe, there is what we call, there are two membranes. These are called pleural membranes. Pleural membranes. And between these two membranes, there is a pleural cavity which is filled by a pleural fluid. Now, these pleural membranes have three basic roles. One, to protect the lungs. But secondly, stop leaking. They stop leaking of respiratory gases into the thoracic cavity. Gases into the thoracic cavity, but also they reduce friction. To reduce friction between the lungs themselves and the thoracic cage. That is, as I have said, that the surface of the lung, each lung lobe, is overlaid by two membranes, the outer pleural membrane and the inner pleural membrane. Between the two membranes, there is a pleural cavity, and within this cavity, there is a fluid, and this is called pleural fluid. And the pleural membranes, among others, we have seen that there are three basic functions. One, to protect the lungs, as they are, of course, extend structures. But secondly, they prevent leakage of gases into the thoracic cavity. And the third one is to reduce friction between the lungs themselves and the thoracic cage. My dear student, we are breaking shortly. When we come back, we shall continue with the structure of the lungs. Now, in the case of any question, any query, please, you may send your normal short messages, a normal SMS, or you may send your message through WhatsApp. You may record and send us your voice note or a video asking a question, giving a suggestion, and whatever. The number that you will use is 0769 04 85 94. I repeat, 0769 04 85 94. Or you can follow us on an Instagram page, that is Elimika Darasa. Follow us there, or you can go in our YouTube channel, that is Elimika. Subscribe and like. After this short break, we will be back again to continue with our program. So please stay around, don't go away.